Here we have the debate, pros and cons of decentralization of CAR T cell production. For the pros, we have Dr. Saad Kenderian, and for the cons, we have Dr. Jacobson, who have also already been introduced. But we can also look at this debate in another way. This is a debate between the Mayo Clinic, which is the #1 hospital in the world, as per Newsweek this week, consistently, versus Dana-Farber, which is part of Harvard, which is the #1 university in the world. It's also a debate between delivered pizza versus your mom's home-cooked food, right? And I think Dr. Shah alluded to this as well, right? Okay. So what would you rather have? We didn't plan it this way, but no pressure on you, Saad. Okay. So this will not be the format of the debate. We didn't have Wi-Fi in our house yesterday, so my son was playing with these stick figures on computers, so I asked him to make this, and he kindly did, and sent it this morning. So the actual format of the debate will be more like this. So that's my introduction. So we have Dr. Saad, no more than 10 minutes to talk about the pros of in-house CAR T production. Dr. Jacobson can present the cons. Then we'll have give Dr. Kenderian 5 minutes to rebuttal any points made, and then we'll open the floor for questions. Now we really have to stick to time, so I'm going to time you. The time now is 3:49. Oh, sorry, 3:49.
Thank you very much for the introduction. And it was really a last-minute debate. We changed the topic of the debate just literally a few hours ago. So before I start on the advantages of in-house versus centralized manufacturing, I want to go briefly into the history of CAR T. And yeah, these slides are. So we don't have a specific slides for the presentation. It's just an open format between us and Dr. Jacobson. So if we want to go back into the history of CAR T, how CAR T cells developed. So I think it was discussed here that the concept of CAR T cell has been old. It's been in academia since 1989, trying to do R&D to improve it, to get it to work in animal models and then to go into human studies. And it's also important to note that the first CAR T products were in academia. The first clinical trial was conducted at the University of Pennsylvania at the NIH, included a few patients. Following that, there was larger clinical trials, again conducted in academia. And in 2013, Novartis partnered with the University of Pennsylvania to take the product to the market. At the same time, Kite was launched and partnered with NIH to take their product to the market. And Juno was launched and partnered with Memorial Sloan Kettering and with Fred Hutch to take their products to the market. So let's go on what's involved in CAR T cell therapy in manufacturing. There are 3 components. There are 3 regulatory GMP requirements. First, you need the plasmid. That's a DNA that dictates what CAR and what's going to be inserted into the cell. And that DNA needs to be a GMP, a clinical grade DNA. It needs to be made in special manufacturing plants. Second, you need the virus. Now that DNA, you took that DNA, you're gonna make a virus out of that, at least in the FDA approved and the effective CAR T trial today. As was discussed earlier by Dr. Shah, that there are other products that are non-viral, that probably you don't need the virus, but today you need the virus. So you need a GMP grade virus as a second part. And third is making the cells. And these cells need to be made in a clinical grade, GMP grade facility, either at the academic center or in a plant. The manufacturing of the cells is heavily regulated because it's individual. And if you ask what pharmaceutical companies brought to academia, they really brought the infrastructure to regulate the CAR. And that's how they were able to go into a larger multi-center clinical trials and eventually to the market. So now it's FDA approved. What are the disadvantages? So as was shown earlier by Dr. Hashmi, CAR-T is most effective in acute lymphoblastic leukemia, right? But if you ask in patients that are eligible for CAR-T, what percentage in kids, what percentage of patients are actually receiving CAR-T in the world? Any guess? Who thinks it's 50%? Who thinks it's 10%? Who thinks it's less than 10%? It is. It is actually 10% of the kids in the world that need CAR-T receive CAR-T. And the reason it's all of the above. It's the complexity that comes with it, it's the cost, it's the access. That's one problem. The other problem is, as we know, it takes weeks to manufacture CAR-T, right? In the best case scenario, it takes 3 weeks. With some manufacturer, it's taken 6 weeks. And there are challenges. And if a challenge comes at the plant, then the whole manufacturer is shut down. And third, as you innovate in CAR-T, so I run a CAR-T program. If I have a new product that I want to get into patients, if we don't have that infrastructure to make the GMP grade CAR-T, then I'm going to need to find a pharmaceutical company that is willing to work with us to take this product and manufacture it and go into patients. So what is the solution? What can we do if we take the manufacturing and we move this manufacturing from industry to academic centers? What are the challenges? So one big challenge is, so remember, there are 3 parts, 3 GMP parts. These are still the cells that we're going to move. We still need the virus. We still need the plasmid, the DNA. Whether the virus and plasmid can also be made at the academic side is questionable at this time. That being said, University of Pennsylvania, they've done that. Fred Hutch done that. Few centers that have manufacturing of the 3 components. You can outsource it to companies that can make the GMP grade DNA and the GMP grade plasmid for you. But also it can be an opportunity that you collaborate with companies, with CAR-T companies. CAR-T companies own the intellectual property around also the plasmid and the DNA. But instead of getting the cells from them, you can potentially buy the cells from them. So that's one challenge. The other challenge is having the infrastructure to do the cells in-house. And as was discussed earlier, there are systems such as the Prodigy that is automated, and there are several other systems. The Prodigy is made by Multani, is more advanced, but there are several other companies that are as good or could eventually be better. But also there are centers that have GMP grade manufacturing, cell manufacturing. All the transplant centers have some sort of GMP grade manufacturing that they could incorporate the cell manufacturing processes. But what's that? What do we need to get there? We need a process that is compliant with the FDA, and we need a body that regulates this process. That body can be the FDA, but if the FDA regulates the process at each center, then that will be specific to the clinical trial that is conducted in the center. If this is going to be a universal process, the FDA does not approve a process. The FDA approves a drug. Then there should be a third-party regulatory body, a society, whether it's ASH, whether it's BMT, whether it's an international BMT society, that regulates, whether it's FACT, that regulates the production of the cells across the different centers. And part of this infrastructure, we need an investment from the academic centers. So ideally, there should be a mechanism that allows the NIH or other foundations to help the academic centers to establish this infrastructure. So I think, in summary, I think the manufacturing part at the academic center is feasible. Getting the virus and the DNA at the academic center is probably more challenging, at least at this stage. And that's where there could be partnership with companies to manufacture the cells at the academic center. The advantage for that is that the time is going to be much shorter. It's on site. So the average time when we manufacture it in-house is 6 days compared to 3 weeks, 4 weeks. The other advantage is allow the academia to innovate. So if each academic center has a manufacturing, then you can move products from the lab into the clinic faster than trying to work it through which company you're working with and trying to get the company to agree to take that product. And then the third, if this is allowed on a clinical grade product, if instead of CAR-19 being made in at the main companies, if it's made at the manufacturing, at the academic site, is going to be much cheaper and much more accessible to our patients. I think to need that, the big major issue is to establish the infrastructure that controls the regulatory requirements of the FDA and other regulatory bodies of the in-house manufacturing. I think I'll stop here.
All right.
So it's over to you, Dr. Jacobson, and you have 15 minutes to make your case.
Okay, I may lose this debate in my first opening statement because I also agree that that there is a downside to moving this technology out of the academic centers and into industry, and there are all the things that were just discussed. I mean, the CD19 CARs all started as being manufactured in-house at academic centers. The problem is that to run that initial clinical trial to show efficacy and proof of concept in a first-in-human study costs tens of millions of dollars, right? And so that's a huge investment for an academic center to make in a product that they don't know whether it's going to be successful or not. Thankfully, with the CD19 CARs at both at Memorial Sloan Kettering, at the Hutch, at University of Pennsylvania, at NCI, this was successful. And so they were able to publish that initial data to present it and then garner interest from a company to pick up the tab to run the registration study. That would be a nearly impossible task for an academic center to accomplish, both to fund a study like that, and then also to manufacture for 100 to up to 300 patients, as was done on the Transcend study. And so I think that there are, I don't disagree that that being able to keep CAR T-cell manufacturing within the academic center and providing the infrastructure for that is, I don't disagree that that should be our goal. What I disagree with, I guess, is just is how close we are, or how likely it is that we're going to be able to achieve that goal. In short, I think we have some really difficult challenges. So first is, let's say the University of Pennsylvania created their, the predecessor to Tisagen-Leclusel, and they have their manufacturing procedures that are outlined. If they want to now open that in a multi-center study, how is there a mechanism whereby other centers can make their CAR T cells in their own backyards under the same processes and mechanisms that the University of Pennsylvania put into place, without sort of reproducing another first-in-human clinical trial with their manufacturing process? I would argue that to date we don't have a mechanism under our regulatory body approvals to do that. So that is the first challenge. So I think what needs to be put into place is that the regulatory agencies need to have a mechanism to say, we see safety and efficacy at one center, and if you can show us that you can manufacture with similar reproducibility and efficiency, and make products that are in specifications that were set forth by Medical Center A, that you can then manufacture on this clinical trial. But the second is, how do we fund a clinical trial that's going to treat 100 patients with these trials? These are incredibly expensive trials, and unfortunately, right now, the way I see it is that we do need partnership with industry in order to support those trials. The third thing is that our partnership with industry has been fruitful. I think that that this has to. We've already mentioned that for many centers, the plasmid and the vector requires industry support, and then I think there are other academic exercises where the industry and academic centers are able to partner, and I think that helps move innovation. So the threat, I think, to sort of decentralizing all of manufacturing is that we remove industry from the equation, and I think that will to some extent slow down and potentially inhibit some of the innovation. So I think I totally agree that we could do this more, we could do this faster, we could do this more inexpensively, and we could increase access if we were able to do this in academic centers. I just think the path to being able to achieve that right now is really paved with a number of hurdles that it doesn't seem really very transparent to me how we can overcome them. But I'm totally committed to trying to overcome them and trying to work with regulatory agencies to think outside the box and think of a new sort of regulatory path to consider these therapies. I think we have we certainly have examples of this in transplant, and so we can work with what we have in transplant and refine it for cell therapies. I just think that I just think we're a little bit removed from being able to get there, and I don't necessarily see how we move from product X and 10 patients to product X being manufactured in a decentralized manner to 100 patients without a partnership with industry, primarily because I don't know how that gets funded. I think I did under time, so I saved you some time and for having gone over in my talk.
Okay, you did. Thank you very much. Now, so would Dr. Kenderian, if you would like to respond to that, so how would you, in practice, deal with the funding of a it'll have to be a trial like this, and how would you be able to harmonize standards between different centers? Like, how could you guarantee that Center A and Center B are offering the same treatment?
Yeah, thank you. So I agree with the points. I think the regulations of manufacturing, decentralized manufacturing is the main challenge, and that's where we need a regulatory body to regulate this. We've had discussions with regulatory bodies, and as I mentioned earlier, that the FDA does not approve a process, but the FDA can approve facts to regulate the manufacturing. I agree that it is not something that's going to be happening soon, where all centers can manufacture a clinical grade product, but I firmly believe that we need to start working on processes, start working with a regulatory body, and try to involve academic centers in this process. Now, as far as funding for clinical trials, I see that point too. We take CAR-19, for example. CAR-19 was licensed out of academia into industry, and now it's FDA-approved product. If there is a process, and so funding for that has come from industry, right? If there is a process for us to generate CAR-19 using the same vector, and that process is approved, the regulatory process is approved by FDA, by a regulatory body, then that industry partner can, instead of providing the cells, can provide us the vector.
Which you have to buy.
We have to buy, that's correct. But we're still talking about less than half of the cost, a fraction of the cost of what CAR-19 costs now. And I think for manufacturers, selling the vector is an easier process than actually selling the cell, because the investment in selling the cell and the infrastructure and the cost of good is much higher. So I don't agree that, I don't argue that we don't need the industry partners during this process. We need them for the virus, we need them for the DNA, and we need them for the for running large clinical trials. But what I'm proposing is, instead of making the cells, making just the cell part in the industry, is to make the cells at the academic centers.
If I can pose a question to Dr. Jacobson. Now, when we do transplants, whether it's allogeneic or autologous transplants, every patient is batched and dosed differently, whether it's in the dose of the CD34 or whether it's total nucleated cells or whether it's CD3 or other cells, the composition of which we don't even check. Every patient gets something different. With the passage of time, might we see something similar happening with CAR T cells? If you're, as long as you're within a range, that you have a target range of so many transduced cells and so much expression of your CAR, et cetera, and you can show that's a fact, then you're okay. Will that sort of overcome the issue of differences between centers and in the issue of harmonization if we treat it like we treat our standard transplants?
Oh, I absolutely agree, and I think that that's why I made the point that I think we have a pathway paved with transplant to work with the regulatory agencies to figure out a path to approval of a process, exactly what has been referred to before, and not just the product but the actual process. And so that if we can if a center can prove that they can make X number of products within specification, they can be licensed to go ahead and produce that product without having gone through their own first in human phase 1 clinical trial, and that as long as the products that get released from the machine fall within a certain specifications, that those are usable products. I absolutely think that that's the direction that we need to go. I think my concern about sort of taking away. So I guess I have two concerns because of what I think really is a continued reliance on industry in order to support the development of new and novel products. Is that part of the, fortunately or unfortunately in sort of a capitalist society, what motivates the companies to partner is profit margins, right? And so I think that and the way you maintain profit margins and in order to invest in future R&D and continued development and running of a company is by charging more for your product. So that even if we were able to keep this with, if we had a partnership, if we maintained the necessary partnership with academics, with industry partners, and even in order to continue to manufacture these in a decentralized, these products in a decentralized process, the concern I have is that it won't end up being as inexpensive as we really think it could be, because there will still be a markup in order to maintain those necessary relationships. And so I think I don't see a obvious solution to that issue. There still is a reliance on industry, and I think that will sort of hinder how sort of accessible and inexpensive we could make these products for our patients.
Okay, if we can maybe, if you don't have any comments, if we can open the floor or invite some questions from the floor. I don't know if Keith Wonnacott is still here, who gave the earlier talk about regulation, but I don't see him. Oh, yes, he is. But I don't know if he was here for most of the discussion. Were you? Yeah, you might. Putting on your hat from your FDA days, what might your views on this be?
Yeah, so CAR T is just one of many cell therapies that could benefit from decentralized manufacturing paradigm. It's a really difficult task as a regulator to regulate academic centers, because what do academic centers do really well? They innovate really well. What do they struggle with? And I can say this from my experience, maybe others are better than me, but what do they tend not to do so well is do the same thing, exactly the same every time, over and over, in a boring, routine fashion. So deliver your McDonald's hamburger exactly the same way everywhere you go all around the world, right? That's not what academic centers do well. They like to innovate, and so that's the first challenge, is trying to do something that leverages the expertise of the center. But I think the second, from a regulatory point of view, that you know, trying to meet the requirements of validation, having sites develop manufacturing processes and GMPs, and have staff that are trained. It just becomes super hard for academic centers to do that, because it's very expensive, and then and so do we have some examples where things like that have worked? The only example that is close is cord blood. In the United States, the FDA regulates the storage of umbilical cord blood that's used for transplantation purposes. And most of the cord blood collection sites are academic centers. And so...
Sorry, that's my timer.
Okay, my time's up.
So that's fine. You were talking cord blood.
So I'm just... there is sort of a paradigm where it's working a little bit, but I think that's more akin to stem cell transplantation, where you know, it's not an FDA regulated product typically. So anyways, those are some thoughts.
Okay, Dr. Qasim from UCL. Or you can use that one that Saad.
So I think the comments have been very interesting, and the debate's been very interesting. So as I mentioned, at UCL, we're very fortunate to have 4 academic GMP centers. But I personally come from a biomanufacturing background, and for me, building on what Keith mentioned earlier, which is the fundamental approach with GMP is consistently doing the same thing again and again and again. And academic centers, as we know, like to do things slightly differently, innovate. But I think there's fundamental manufacturing issues we need to take into account. The first being the pace at which the technology is changing. So at the moment, as an example, we're using viral vectors. In 5-10 years' time, it's highly unlikely we'll be using viral vectors. So therefore, we need to move to a non-viral approach. How do academic centers take into account the amount of work needed for process change and demonstrating process comparability? That is a huge burden, particularly in a decentralized context where you are going to need to demonstrate comparability between multiple centers for each process change. I think the second thing is implementing, so not only implementing new technologies, but the scalability. One of the key challenges, I mean, we've seen, you know, the Prodigy is a good technology, but the scalability of the Prodigy is highly challenging. It's a single machine which can manufacture a single patient cells, and for about 14 days, it's a very expensive cell incubator. So being able to manufacture multiple patients in parallel is a key issue that the industry is now facing, particularly for autologous therapy. So for an academic center to take on that burden and that cost, I think is a major issue, particularly when we now see the change in technologies and the innovations occurring. So from my perspective, I think the academic centers do a fantastic job, particularly the GMP ones, of innovation and small-scale clinical trials. But I think there will inevitably need to be engagement with industry moving forward, particularly because the costs just become unbearable and the amount of focus on manufacturing is extensive. I mean, even established CMOs are struggling to manufacture cell and gene therapies, you know, regulated and at a cost that is effective. The other point you mentioned on the cost about they are very expensive now, but that is also because many of these companies are recouping R&D costs in the current generation of cell and gene therapies, and it's expected over time, especially if we move to allogeneic, we will get economies of scale as we move to industry. So, but I do feel that there is a major issue around chain of custody as well, which we need to take into account. It's better at academic centers. There's fewer chain of custody requirements, but that is also an issue for the debate. So I think that there is lots of manufacturing nuance needed, but I do think that there is scope for decentralized, whether it's academically led or industry led is another question.
Okay, one last comment from Saad, and then we'll close the session and move on to the last session.
I think all good points by Keith and by the other speakers. I think one point I don't agree with is that the cost is going to go down. I think we have experience with other drugs. When the first generation pill comes on the market, it costs $10,000 a month, and then we're saying it's new and innovative, and we wait for the second generation, and then next generation pill comes on the market and it's $15,000 a month. I think I find it quite unlikely. And I think with gene therapy, the first gene therapy that was approved was Kymriah, and that cost was nearly $500,000. But then after that, we had gene therapy for Alzheimer's, for spinal muscular atrophy, and we'll continue to have other gene therapies. I agree with the regulations. I think the change is also an important point, change of manufacturing, change in the platform. But similarly, if we're making non-viral CAR Ts, there could be a process that is adapted at the academic centers and applied to the future products. I think that goes back to the point that we need a regulatory body that regulates the process and get that process approved and different academic centers licensed to have certain that manufacturing. I mean, I also agree that we need the industry partners in running large clinical trials and providing the other components of the CAR beyond the cell manufacturing.
Okay, if there aren't any pressing points, we'll move on. Thank you very much. Thank you.
Thank you.